What is molecular docking? Docking is very important in biological science and drug action design. Docking is an attempt to find the best matching between two molecules. Two molecules means one molecule's ligand, another is protein or protein, or any molecules what you have synthesized in your laboratory and with the protein. A more serious definition is docking is methods which predict the preferred orientation of one ligand bond in an active site to form a stable complex. You can see here, this is the protein and the ligand. Docking of a small molecule ligand, which is in brown color, with a protein receptor, which is in green color, to produce a complex. What is here theory? Lock and key. Finding the correct relative orientation of the key which will open up the lock. On the surface of the lock is the key hole. You know, in which direction to turn the key after it is inserted. That is the aim. So you have the key, you have the space where you are putting the your key, and you have the lock, and you have the direction. So four things keep in your mind. Now the problem can be thought of as the lock, and the ligand can be thought of the as a key. So you can see here the NVP, the crystallographic structure of HIV1 reverse transactions. Green colored P51 subunit and red color P66 subunit. So each color have their own significance. Here is the key or ligand. Now molecular docking. The aim to achieve an optimized conformation for both receptor and ligand, and relative orientation between protein and ligand such that the free energy of the system is minimized. This is the target. Successful docking means methods search high dimensional space, effective and use a scoring function and correctly rank candidate docking. You can see here the target, that is your protein. This protein you can easily obtain from the protein database. You have the ligand. This ligand you can easily find out from any research paper or PubChem, or you have the crystal structure, or you have the draw structure of your new molecules. And then after docking, you can see the molecular docking complex. Now, friends, what is the importance? Molecular docking, rational design of drugs. That is the main aim. Identification of the ligand correct binding geometry. That is the pose in general language. In the binding site, predictions of the binding affinity scoring function. My friend, types of docking. Rigid docking, lock and key. Lock is rigid, key is rigid. So in rigid docking, the internal geometry of both the receptor and the ligand are treated as rigid. Flexible docking induce fit, and enumerations on the rotations of one of the molecules, usually a smaller one, is performed Every rotation, the energy is calculated. Later, the most optimized pose is selected. So, docking can be between protein ligand, protein protein, and protein nucleotide. Here, new, don't I think you don't know, protein nucleotide docking is also possible. Now, you can see in this figure protein protein interactions, protein ligand interactions, and protein and nucleotide. You can see the DNA, protein, and nucleotide. Now, type of interactions: electrostatic force, electrodynamic force, steric force, solvent related force. These forces are important. And what is the electrostatic force? You know, the forces with electrostatic origin are due to the change resulting in this matter. Electrodynamic forces, the most widely known is probably the van der Waals interaction, you know it. Steric forces, these are caused by the entropy. For example, in cases where entropy is limited, there may be forces to minimize the free energy of the system. That is the steric force. And solvent related forces. These are due to the structural change of the solvent, and these structural change are generated when iron, colloids, proteins, etc. are added into the structure of the solvent. The most commonly as hydrogen bond and hydrophobic interaction. Friends, key stages in docking. Keep in your mind, key is your ligand. So, what is the key stages in docking? Target, receptor selections, and preparation. You cannot uh, open your all lock with the one key, but nowadays this system is also developed by the engineers. So, ligand selections and preparations, docking, and evaluating docking results. Now, a typical docking workflow you can see here. Now, target selection, ligand preparations, target preparation, docking, and evaluating docking results. Receptor selections and preparation. How you will do the receptor selection? You know, keep in your mind, receptor is your protein, your buildings, and the receptor. The 3D structure of the receptor should be considered, which can be downloaded from PDB. PDB means protein database. The available structure should be processed. The receptors should be biologically active and stable. Identification of active site. How you will identify the place. The active site within the receptor should be identified. The receptor may have many active sites, but the one of the interest should be selected. Ligand selections and preparation. Ligand can be obtained from the various databases like Zinc, PubChem, and other sources. You can draw it, say a sketch using tools like GameSketch. Now, docking. The ligand is docked onto the receptor and the interactions are checked. The scoring functions generates score depending on which the best fit ligand is selected. Now I am talking about the software. So a lot of software, plethora of software used for the docking. Some of them I have here. I have to mention it. Sanjeevani, IIT Delhi. You can go through this website. Gold, University of Cambridge. You can go through this site. Autodoc, Scripps Research Institute USA. JMDoc, uh, like that Higgs protein docking, University of. So you can visit this all website. You will find grams global range molecular machine matching. That is the protein docking. Now the application, which is our important target. Why we are doing this? What is the important of docking? So virtual screening, heat identification, docking with a scoring functions can be used to quickly screen database of protein drugs in silico to identify molecules that are bind to protein target of interest. And drug discovery, that is the late optimization. Docking can be used to predict in where and in which relative orientation a ligand bind to protein, binding mode or pose. This information may in turn be used to design more potent and selective analogs. Means your small molecules is drug. If it is working properly with a particular protein, then these molecules may be a good candidate for the drugs. Now, bioremediation. Protein ligand docking can also be used to predict pollutants and that can be degraded by enzymes. Now, these are the references and other references where I have taken this PPT. So, this is only for the education purpose. Thanks for watching.